then we're going to be tired all the time, not having any energy. Now, one thing to point out here, and I'm not doing this to persuade you toward a certain kind of diet, but particularly a vegan diet. People who want a vegan diet need to be aware that their diet may not be supplying certain things that they need. There's ways, of course, to supplement that, but not everybody is necessarily aware of it. And iron would be one of those things, and also potentially calcium. Although there are a variety of foods that have these things in them, just maybe not as in a rich supply as animal products. Iron being one of them. So we have heme iron versus non-heme iron. Now, heme iron would mean a flesh diet. Because red meat, for example, which we've been advised perhaps is not the most helpful because of the increased risk, for one thing, for colon cancer, but nevertheless, there are a lot of people who like their steaks and whatever that has heme in it. Now, heme is a big molecule. So how do you get this thing across in enterocyte? Well, it takes a special transporter to do that. But there is a transporter that will move the heme molecule into the enterocyte, and then it gets broken down. Now, the breakdown of heme is typically like that it happens in the spleen. You're going to get some biliverdin, which is toxic, and you're going to get some carbon monoxide. And remember, this has something to do with oxygen transport. So that's an interesting relationship that we have there. So the heme molecule can be broken down, and the iron is released, and we can absorb it. Now, one of the things you'll notice here is there are a number of iron-binding proteins. Ferritin is one. Mobile-ferrin, MF, mobile-ferrin. This IREG, that's another iron-binding protein. Transferrin is another one. So we have a number of iron-binding proteins, and you don't have to exert too much imagination to see that if we're deficient in any of these iron-binding proteins, we've probably got a problem. And potentially there's some rare situations where that could occur. So we can extract iron from heme and get it into blood. It binds to transferrin, and it will go to a storage site like what? The liver, or to a utilization site like bone marrow, so we can make red cells from it. But apically, we have also another mechanism for absorbing iron, and this is ferric iron, Fe+3. Now we can't, for some strange reason, the enterocyte cannot absorb ferric iron. But the membrane, the apical membrane, has a reductase that will reduce the F3 to 2, and then you can absorb it. Now keep in mind, this is an apical membrane protein that can do this. So, normally our enterocytes are equipped to handle ferric iron in that form, ionized iron in that form. But it's handled different than heme iron. Even though it's the iron that we're really after, they're absorbed apically quite differently. So there's a special dication transporter, DCT1, that transports iron Fe2 with hydrogen ions. Now this is a unique transporter because so much of the time when we talk about active transport, we're talking about sodium. This is not sodium here. This is hydrogen ion. But this is a co-transport process. So the ferric iron has to be reduced to ferrous iron, which can then be absorbed and then combined in these proteins and then end up in the blood, just like the iron from heme can do. So this is a tremendously oversimplified depiction of the absorption of iron, because it's really a lot more complicated than that. Now another one is cobalamin, or B12. Now why is this important? Well, at the very bottom of this chart you see that it decreases, absorption of B12 decreases with age. It's a very, very important vitamin, even though you do not need much of it. It's very important neurologically, and it's also important to prevent severe anemia. So people who have a severe B12 deficiency need to supplement their diet. And when you have blood drawn, they can test for this. They can determine whether you're B12 deficient or not. So if you're young like you are, this problem, not concerned about this. But once again, the best source is animal protein, not a vegan diet. So we have calcium, iron, and now B12, which are in greater amounts in an animal protein kind of diet. Now this is a bit complicated in the way it's handled. There is a substance, there's a protein called haptocorin that binds cobalamin at low pH. Now where would that be? Well, that would be in the stomach. So at low pH in the stomach, cobalamin binds haptocorin. Why does it do that? Because the low pH will destroy the cobalamin without this intervention. Now proteases will hydrolyze, will break this complex down, but now the B12, the cobalamin is vulnerable again. So another factor, the intrinsic factor, abbreviated IF, binds the cobalamin. So first haptocorin binds it, protects it against low pH. Intrinsic factor binds it to protect it against it being broken down, digested. But notice again that the ileum is playing a very important role here because the receptors that allow for the absorption of cobalamin are found in the ileum. So again, we're kind of building a series of little scenarios where the ileum is important for absorption, even though you've got all the rest of the small intestine involved. And we really don't know how this complex is absorbed into blood, hence the question mark, or into its target cells. We don't know what that transport is, but in the blood itself, transcobalamin 2 is carrying it in that form. So it's a bound form. So a lot of these substances, like calcium, like iron, are bound to something and transported that way, and cobalamin is essentially no exception. Now let's move on to how we handle dietary carbohydrate and then protein and then fat. So we know about amylase. We know that saliva has amylase in it, so starch digestion begins in your mouth. The more slowly you chew, the better digestion will occur. It's not complete typically because the low pH of the stomach will stop any amylase activity. But once the contents of the meal have been emptied into the duodenum and exposed to pancreatic fluid, then we've got amylase again. So when we talk about starches, for example, being involved in being digested, we're talking about salivary amylase and pancreatic amylase. And the end products are some simpler ones, including glucose, but maltose and alpha-dextrin cannot become glucose without an enzyme bound to the apical membrane. Now this is kind of interesting because what this is saying is that we've got enzymes that are part of the enterocyte that are required for handling starch that's essential, also essential just like amylase is. So if we don't have these proteins, these enzymes in the apical membrane, we're not going to handle starch very well. Now lactose, another a disaccharide, and sucrose. So we've got maltose can be a product, it can be absorbed as glucose only because by having maltase. Lactose, in the apical membrane, there's an enzyme that breaks down lactose into galactose and glucose. That we can absorb. We do not absorb disaccharides. We absorb monosaccharides. Then there's sucrose that can be handled, very common sugar that's added to all kinds of things. There is an enzyme, apical membrane, that breaks down sucrose, so we call it sucrase, ASE for enzyme designation. So sucrase breaks down sucrose into fructose and glucose, and we can absorb those sugars, and ultimately they go into blood. We'll look at that just a bit in a moment. Now, what is lactose intolerance? Have you ever heard of anyone who has lactose intolerance? It's much more common in Asians than in other groups. I just assume it's a genetic thing. People who have lactose intolerance have difficulty when they drink milk or eat ice cream or anything that has dairy products in it. Why? Well, it's because they either have inactive lactase. Lactose is coming from the milk sugar. So they either have a defective lactase, or maybe there's no lactase at all, and the severity of the lactose intolerance would be related to, what the problem actually is. But basically, it has something to do with lactase, the inability to digest milk sugar. So what do you do about it? Well, we'll come back to that in a minute, because the question is here, when you talk about malabsorption, have lactose intolerance, what are the symptoms? Well, bloatiness, that discomfort associated with that, poor digestive processes, of course, anything having to do with sugar, excess gas. These are symptoms. There are probably others that go along with it. So what do you do about it? Well, just leave dairy products out of your diet. That's a simple way to do it. And there's varying aspects of it. Some of it can be much more serious than others. Okay, these simple sugars, remember we do not absorb complex sugars. We only absorb the simple ones. So that means that we have to have these enzymes to break down the disaccharides into monosaccharides, because only the monosaccharides can be absorbed. So here we see glucose and galactose co-transported with sodium. Now this is that transporter that I mentioned earlier that favors the absorption of water, because water tends to follow the absorption of sodium and glucose, because they have, and galactose, because they have osmotic effect. So that's how glucose and galactose gets into the cell. Fructose has its own separate transporter. I don't know why, but it has its own separate transporter. But there is a common transporter basolaterally that will move any of these simple sugars into your bloodstream. And the reason why sodium-potassium exchange is important is because this transporter cannot function without it. Because any co-transport process that involves sodium has to have sodium-potassium exchange operating at the same time. You know why? Because this is operating by way of a gradient. And this favors, the sodium gradient is favoring this transporter, and the only reason we have a gradient is because sodium is being moved out of the cell. That generates the gradient. So that's a basic thing to remember, is that co-transport with sodium does depend upon sodium-potassium exchange, regardless, essentially, of what it is. Now, there's another interesting situation that some people have, and that's gluten intolerance. This is an interesting one because, for whatever reason, and I guess you could call it an allergic response to wheat gluten. And it's pretty hard to find. It's somewhat of a challenge to find foods, these certain foods like cereals and bread and so forth, crackers and so forth, that don't have gluten in it. And yet gluten intolerance can be problematic. The symptoms of gluten intolerance, again, are excess gas, poor digestion, bloatiness, these same discomforts associated with it. But there's something about the gluten that actually causes damage to the villi. So here we see normal villi, the little projections, and the enterocytes that surround them and so forth. They're small, they're microscopic, but nevertheless they're prominent. So what does gluten do? It damages the villi. It causes the villi to be stubby. And why is this important? And I didn't mention this before, but most of the absorption occurs in the upper third of the villus. So if you're losing height in terms of the villus, you are impairing absorption and anything else that's taking place in the upper part of the villus. So what do you do about this? Well, again, leave out of your diet foods that contain gluten. And you can develop allergy reactions to all kinds of things, including gluten intolerance. But lactose intolerance, gluten enteropathy, or gluten-induced celiac disease, as it's called, are interesting because they're diet related, and there are people who have these problems. Any question? Okay, let's move on to protein. Here we have a number of proteins that are involved in digesting proteins. We get oligopeptides, which are just very small proteins. We have the di and tripeptides that, this links is 2 or 3 amino acids, then single amino acids. So what do we absorb? Well, we can absorb di and tripeptides. We can also absorb amino acids. This is apical side. This is basolateral here. Okay. Now, another membrane-associated relationship, an important one, is that in addition to having enzymes that help us digest sugar, carbohydrates, we also have enzymes, membrane-bound, apical membrane-bound enzymes that allow us to handle protein. And so the pepsin, the proteases from the pancreas and the pepsin from the stomach, those are beneficial, but they're not enough. And so we have these membrane-bound enzymes that also play a role in the absorption. Now the transporters that we're talking about here for amino acids, we said di and tripeptides can also be absorbed, but usually it's single amino acid. It isn't shown here, but this can get rather complicated as we look at, again, a little bit different perspective. Here is the transporter for di and tripeptides. And the transporter for amino acids on the apical side, probably the most common one is classified as B. I don't know what the B is for, but this is a transporter for single amino acids. And notice that through this transporter, these are co-transported with sodium. So what's going to follow? Water. So we've said that the co-transport of simple sugars with sodium, co-transport of amino acids with sodium, are going to tend to pull water with them. Now the amino acids that are, the transporters for the amino acids or the di and tripeptides on the basolateral side are different. It's very complicated. There's probably 6 or 8 different amino acid transporters. On the apical side, this is the most common one, but there's many, many others. So that means that an individual who's deficient in these transporters are going to have problems with the absorption of amino acids. So the key points here are that the apical enzymes are essential, and that's shown in this slide. The apical enzymes are essential to being able to handle amino acids, and then we have different transporters for them, basolateral and as well as apical. What about fat? This substance is significant in most of our diets. Fat is significant, but the carbon chain length does have something to do with it. That is to say, when you have fats, remember from biochemistry, or from organic chemistry, you have a glycerol molecule with 3 carbons, and then to each carbon there's a fatty acid. The carbon chain length of the fatty acid is critical, not the 3 carbons in the glycerol. So when we're talking about long carbon chain length, we're talking about maybe 10, 12, or more carbons in the fatty acid chain. So they're handled differently. For example, if we look at glycerol, which is the molecule without the fatty acids, it's absorbed in its own unique fashion. If we're talking about a long carbon chain length fatty acid, or triglyceride, then we've got a different situation. If the long carbon chains of the fatty acids are separated out from glycerol, and glycerol can be absorbed, as I said, independently, or is absorbed independently, but the long carbon chain length fatty acids, or monoglycerides, are incorporated into a little particle called the micelle. Now these are microscopic, but they are, it's a small package because there's all kinds of things in it. Notice that conjugated bile salts or acids are critical to the formation of it. So going back to our discussion of bile secretion and bile acid synthesis and all of that stuff, this is where it comes into play. Without the bile acids, you can't form micelles, which are critical to the way your body handles fat. So the reason why we recycle bile acids is all linked to fat in our diet. So what happens is that these micelles get taken up by enterocytes and broken down into some component features. Now there's a lot of other things that can be in this micelle. You see phospholipids, cholesterol, and even some proteins. A lot of things are, and fat, by the way, fat-soluble vitamins and any fat-soluble medication that your patients might be on also gets incorporated into micelles. That's the way your body basically handles them. But what happens when these micelles are, actually what happens is the micelle breaks up. It falls apart. And the components, whatever's in it, gets taken up by the enterocyte. Not the whole micelle, just what's in it. And what happens is that the components that were in the micelle get packaged again, repackaged, as chylomicrons, and the chylomicrons go into the lymphatic system. So the reason why we have lymph vessels in the villi is so that the body is capable of absorbing substances into it, including chylomicron. Now, I don't know if any of you have ever worked in a lab setting where you've been exposed to this. But if you were to take, well, let me quickly back up. Sometimes when you have blood tests done, and especially if they're looking at cholesterol, they're looking for fat in your blood and so forth, you have a fasting time. You're not supposed to eat after midnight. Remember that? The reason is because recent meals can impact things. So if you took a blood sample from someone who had had a high-fat meal, and maybe they went to In-N-Out Burgers and they had a milkshake, the plasma would appear cloudy. Now, why is the plasma cloudy? Because it's loaded with chylomicron. Now, those will dissipate over time, but you have to have several hours for it. So if you're doing a cholesterol test from a blood sample, you don't want to have eaten a few hours before something that contains fat, because you're going to throw this whole thing off. So chylomicrons and micelles are different. They're similar, but they're also different. So let me help you here to distinguish the difference between these two particles. Micelles will have bile salts linked to them. Chylomicrons will not. The bile salts have been recycled. Bile acids have been recycled. So the chylomicron is not exactly the same as a micelle, although there's some definite similarities to it. Now, we've talked about this before, and I don't think there's any reason to spend much time on it again. It's just that when we're talking about the importance of bile acids having to do with how your body handles fat, then that's when these uptake transporters for bile acids and bile pumps, or bile acid pumps, are essentially important. Okay. Most of the water that we run through our bodies is absorbed in the small intestine. About 1.5 liters will enter the colon per day. Now this can change, of course, but we're looking at the final processing of water in our bodies. So under normal circumstances, 1.5 liters enters the colon through the ileocecal sphincter. Almost all of that, maybe 100 mils, is not reabsorbed. So we end up with stool water amounting to about 100 mils. Now, we can change these numbers, and these numbers are sort of arbitrarily designated here, but let's suppose that instead of 1.5 liters, we push through 4 liters. Our colon can accommodate that, such that we still only have about 100 mils of stool water. And we can have problems a couple of different ways. One is that if we overwhelm the colon, if we send too much fluid to it, it can't handle it. And so the 6 liters, it can only absorb a max somewhere, and there's probably some variation in this, but if the max is about 4.5 liters, then the stool water will become well over a liter, and we've got diarrhea. That's one scenario, overwhelming the absorptive capacity of the colon. The other is if there's some inflammatory condition that is causing the colon to secrete. The volume of water coming to it is okay, but it's not absorbing, and instead it's secreting, or let's say another way, it's secreting more than it's absorbing. Then this can also result in excess stool water, and again, another cause for diarrhea. Now, there's some disorders out there that are not that uncommon. One of them is called irritable bowel. And people who have irritable bowel are very subject to stress, and they can cycle in and out of constipation and diarrhea. It's a wonderful way to live, but it's, it happens to people. So a lot of it just has to do with, first of all, most of the water is normally absorbed in the small intestine, maybe as much as 10 liters a day, small intestine. And the colon is the final region where there's sort of some final adjustment to how much water is actually lost. Now let's talk a little bit about bacteria. I don't know if you're well aware of this, probably not, there's no reason why you should be, but there's a lot of interest in gut bacteria, particularly I hesitate to use the word health fanatic, but that's sort of what they are. Get so absorbed in this that they will actually have a fecal sample analyzed to find out what bacteria they have. Now, why is this? Well, because the gut is filled with bacteria, especially the ileum and the colon. But they're there for a reason. We have far more bacteria than we have total cells in our body. There are a lot of them there. Now, there's a lot of interest in this because it's highly possible that the nature of your diet can select for certain bacteria in your gut. High sugar diet being one of them. And because there's connections between your gut and your brain, it can actually impact your desire for certain kinds of food. For example, if you have bacteria that like carbohydrates, then you're going to be stimulated to take in far more sugar in your diet than you probably should. That kind of thing I'm talking about. But these are reasons why the bacteria that we normally have are important. They're involved in a variety of things. Don't feel like you have to memorize this list particularly, but they have to do with detoxification, with some digestion, even with expression of this transporter. And there's other things that are important, importantly related to these bacteria. So therefore, they're there for a reason. Now, what keeps them under control? Somebody tell me. We talked about it. What keeps them, the bacteria population, under control? Fasting motility pattern, right? If you think of a broom sweeping contents from a floor, the fasting motility pattern can be thought of as a broom that is sweeping out the contents, the residual contents of the antrum of the stomach and the small intestine. It goes all the way through to the colon. That's the fasting motility pattern. Phase 3, that's when it's happening. Linked to motile, remember that? Okay. The bacteria in our gut, some of the things that it works on are endogenous, that means produced by the body, and you'll notice that urea is one of these, and you'll notice their primary and secondary bile acids that are deconjugated or dehydroxylated. That's part of what we discussed in the bile acid cycle. So bilirubin is a toxin, of course, it's coming from breakdown of red blood cells, and we have to get rid of it. That bacteria help us do. Then there are exogenous substances, that is coming from outside the body initially, including carbohydrate and fat. And there are short-chain fatty acids generated, hydrogen gas, carbon dioxide, methane even. You know, there's a lot of concern about dairy farms because of all the cows that are producing methane. So there is methane produced by fermentation enzymes that are related to certain things in our diet. These can appear in flatus, or also they can be exhaled in the breath. Amino acids. So there are good things that bacteria are doing. I don't know if you can say that generating methane is a good thing, but nevertheless, it is true. Some of these gases, if you know, particularly these two, are either flammable or explosive. Kind of interesting. Hydrogen sulfide is another one that can appear in flatus as well. So again, don't feel like you need to memorize the table, but just to appreciate that the bacteria that are in our gut are there for a reason. Now, why do we not want overgrowth? And I just asked you a few minutes ago, what is there that prevents the overgrowth? Well, why do we not want overgrowth? Well, because if we do, we're going to have fermentation. That's not good. We can have a B12 deficiency because the bacteria will use B12. It affects the conversion of primary to secondary, and the most efficient bile acid form is primary. Also, it can affect the immune responses and result in inflammatory conditions like IBS or Crohn's disease or some of these other things that are impacted. Now, it's very, very important that that there be a balance between absorption and secretion. I think you can see that. The absorption tends to occur in the upper part of the villus. The secretion tends to occur down here. And the point where we're going with this is that what happens if there's an imbalance? What if there's an imbalance between the absorption, which is up here, and the secretion, which is down here? We can see that because essentially any of these transport mechanisms that we have kind of referred to, like this one with glucose and sodium, or amino acids and sodium, which isn't shown here. Actually, this could be either one. Any of these transport processes, either here or at the tight junction, or maybe even basolateral, inflammation can affect all of those, or any of those, and result in situations where there is no longer a balance between absorption and secretion. And we know what the problem can be, because we said that one of the problems, potential problems, causes of diarrhea is if you have colon secreting more than it's absorbing. The same thing could also happen in the small intestine. It's not absorbing properly. This is an interesting slide. It's not up to date, but every red dot represents 1,000 deaths, and most of them are children. Look where they are. They're mostly, we see quite a bit in especially India, Pakistan, Bangladesh, Africa, regions of Africa. Not so much the United States for some reason. South America, Mexico. That's tragic. That's due to one virus only. Infection with one virus can result in death, gastroenteritis, which is going to include dehydration, and the consequences. Very interesting. Okay, let's talk finally about what regulates food intake. This is an important issue. Strangely enough, half of the world, roughly, is overfed and half is underfed. And we have it in our own inland empire. People, we tend to be more sympathetic, I think, with children than we do adults, but there are hungry people in our neighborhood. Now we're approaching Thanksgiving, and all of these agencies that are involved in feeding homeless people and hungry people are asking for donations, which is okay. But what is it that regulates food intake? You'll notice there's short-term influences and long-term. And most of the long-term impact or controls over food intake have to do with hormones. We'll see how that works. Metering effect. This has to do with chewing and with swallowing. So, the slower you eat, the more you chew, the less you eat. So you can see fast food places don't really encourage this kind of thing. Gastrostatic effect, that's distension of the stomach. When food enters your stomach, it stretches it, and that tends to impact food intake. Thermostatic effect has to do with the change in body temperature. It's probably fairly minor, and nevertheless, metabolism will change body temperature and influence. The glucostatic effect has to do with your blood glucose level. When people are hungry, they've been fasting, what happens to the blood glucose? It goes down. And there's hormones involved. There are GI hormones that are all part of this process. 300,000 deaths are attributed to obesity. It's been increasing in adults as well as children. And when we talk about obesity, we can say overweight, not necessarily defined the same way. It's a worldwide problem. It's not just in the United States, in spite of the fact that we have hunger. This is an interesting map. I'm sorry I don't have the up-to-date one because it's updated about every 10 years. And so the most recent one that I have is 2009. There should be another one out there somewhere. I just don't have it. But the interesting thing is that we're looking at obesity in the context of state-by-state relationship, even including Alaska and Hawaii. And so obesity under these terms are defined as something to do with body mass index, about 30 pounds overweight for somebody of that height. The interesting thing about it is to look at this designation at the bottom according to colors, telling you which state has what percentage of people are obese. Now one of the things to notice here has to do with those that are greater than 30%, and you can see where they are. There's an interesting one here, and that's Colorado. It has the least obesity. Now that is subject to something that, and this has happened elsewhere, with the legalization of marijuana. Because marijuana stimulates appetite. So now you have a state, not the only one, California is included now too, but you have a state, you have low levels of obesity where marijuana is legal, and I would like to see if the incidence of obesity in Colorado has changed. I suspect it has. But that's kind of an aside. So there are, for whatever reason, there are parts of our country where obesity is a greater problem. Now, what is involved in regulating this? Well, you'll notice that vagal reflexes apparently are. That would include distension, stretch of the stomach. When you eat a meal, it has a satiety effect. There are a number of, we have some fat storage. And fat tissue can produce, or does produce, a hormone called leptin, which inhibits the feeding center up here in your brain. Leptin from adipose tissue inhibits the feeding center. Now there are other hormones that are part of the eating process, CCK for example. When you eat fat, you're going to get CCK. CCK has a satiety effect. It inhibits appetite. Also inhibits gastric emptying too, for that matter. Ghrelin is another substance coming from the stomach, and maybe elsewhere, but primarily from the stomach. This is a stimulatory pathway. If you follow the black line up, you will see a plus at the top. So ghrelin from your stomach will stimulate food intake, make you hungry, and it's a factor of fasting. There's some other neurotransmitters like PYY and this involved. Insulin, another one. Insulin also, because it's related to blood sugar, will tend to inhibit the intake of food. So I'm only conveying a portion of the complexity of this, because it would be very convenient if we could say we've got a hunger center up here in the brain, and we've got a satiety center, and all we have to do is shut one down and turn the other one on, or vice versa. It doesn't work that way. These centers, these satiety centers and hunger centers and hypothalamus, do get all kinds of signals, but it's not so simple. Unraveling or solving the obesity problem is not simple, just dealing with these pathways. even though they're important pathways and even though things that are going on, they're important. So in this cartoon of the hypothalamus, we are seeing different kinds of neurons. And I've defined them for you because otherwise you wouldn't know what in the world this is referring to. But you'll notice that one of them, the cart neurons, are related to cocaine and amphetamine. These are kind of pleasure stimulators. So it's interesting that these kinds of stimulators have been linked with food intake. So in this particular region of the hypothalamus, the arcuate nucleus, we have different neurons that are influencing food intake. So this would be the hunger side, and this would be the satiety side, decreased food intake. And they're interacting with other neurons up here in a different part of the hypothalamus that have to do with how much we eat and have to do with metabolism and energy expenditure. So it gets very complicated, and I think this is probably part of the reason why it's been so difficult to find a so-called cure for obesity. It's just not a simple situation whatsoever. So we have neurons, to simplify this as much as we can, we have ghrelin, which can stimulate the feeding center. That would be these neurons. We have insulin, leptin, and CCK, which are inhibiting the feeding center and stimulating the satiety center. So that's how you can fit them together or relate them to each other that way. So, but the other thing that's noticed here, we can notice here, is that even though we're talking about neurons in the feeding center and neurons in the satiety center, they're doing different things. They're interacting with each other. And they're interacting through the release of, because of the presence of certain receptors, like this MCR thing here, and alpha MSH. A bunch of stuff involved in this. We see alpha MSH involved up here, and there's other things. This neuropeptide Y receptors involved, MSH receptors involved. Complicated, no question. And there are genetic predispositions to how this whole system operates, and therefore some individuals, some families have more difficulty with this than others. So to sort of summarize, what is regulating food intake? Gastric distension is important. That's through the vagal reflex. Chewing, changes in blood levels of glucose and amino acids and fatty acids, that's all fed state. So this is all part of why or how our brain gets satiated with respect to food intake. As we know, Thanksgiving is coming up, so most of us are probably going to overindulge in one way or another. So don't allow yourself to get indigestion over Thanksgiving dinner by thinking about this stuff, but it's all going to be involved. So we have stimulating factors for food intake. You can see what they are. Interestingly, we have the endocannabinoids. That, of course, would include the food stimulating impact of the cannabinoids, which, of course, are found in marijuana. Then there are satiety factors. All of these are listed here. So we have a balance of these, and at certain times, these are going to predominate. These effects, and certain times, these are going to predominate. And so the whole process is rather complicated. A bit about fructose. Because of the obesity problem that is ever increasing, fructose has been implicated. There's a lot of research has been done, and some of the information that I have here are proposed mechanisms of what fructose does to you. Now, I know you've seen this, and maybe you've even been a participant. But you can go to In-N-Out, you can go to Del Taco, you can go to any of these places, and you can get a container this big or bigger, a Pepsi Cola, a Coke, or Dr. Pepper, loaded with fructose. Now, can we attribute obesity in prosperous societies to strictly to that? I don't know, but there's more and more who feel like it's contributing to it. So, what do we know about obesity and fructose that might be implicated here? Well, one is it can cause insulin resistance. Now, insulin lowers your blood glucose. So that means that we've lost some control over blood glucose. Another would be that there's more triglycerides circulating around. And of course, triglycerides can be stored as adipose tissue. And this seems to primarily focus on abdominal obesity. Leptin resistance. Now, why is this important? Because leptin is an inhibitor of food intake. It's in the satiety pathway. There can be postprandial, if you're not familiar with that term, this means after the meal. Ghrelin levels that are circulating fail to decline after you've eaten the meal. Now, ghrelin, remember, stimulates food intake, and if it fails to decline, even though you've eaten, that can be problematic. Another is that postprandial leptin and insulin can be impacted as well. So the hormones that we pretty well know are influencing food intake and satiety can be impacted possibly, quite possibly, by how much fructose we are taking in. And food preparation companies have been responding to this. And you'll see fructose-free and reduced sugar. But they put the other things, sweeteners in there that can be problematic as well. But it's kind of interesting. Okay, that ends GI. So we'll have a review, I think, on Tuesday. Is that right? Yeah, I know. Well, the plan is this. Review Tuesday, have the quiz Friday. What? Is that bad? It is bad?
No, that's great.
Okay. Well, that's the plan. Now, it hasn't been solidified because I've taken an initiative that Dr. McLean has not agreed to yet, but that's why we plan. Review Tuesday and quiz first thing Friday, which I think is like an odd hour, 10 o'clock.
I hear it.